In this dirty biochemistry lesson, we're going to continue our discussion and talk about ketone body synthesis. Ketone body synthesis, as an overview, is a process by which ketone bodies are formed from fatty acids in the liver to provide fuel to the body when glucose is otherwise unavailable. It typically occurs when glycogenolysis and gluconeogenesis substrates are both exhausted. And as you'll see later in this video, that typically occurs in periods of starvation. Ketone body synthesis may also occur if the TCA cycle or the Krebs cycle is unable to function. And as you'll see later on in this video again, and this sort of goes with bullet point number 2, this occurs in states such as diabetic ketoacidosis, also known as DKA, and in chronic alcoholism. Now, this is everything that you have already seen in the previous lesson on fatty acid oxidation. And just to review, we need to quickly summarize what we've talked about because that segues beautifully into ketone body synthesis. So everything that you see here in blue is fatty acid oxidation. Again, the fatty acids come all the way down, get converted to fatty acyl CoA, are then converted to acetyl CoA, and once you're at acetyl CoA, you have the option of either directing that toward the TCA cycle for further production of metabolic intermediates and ultimately ATP in the electron transport chain, or by directing it to form ketone bodies through ketone synthesis. So shown here in red is what this video is going to be talking about today. It's how we start with acetyl CoA and ultimately produce ketone bodies. Now, let's get right into the biochemistry of ketone synthesis. So obviously, as I just told you, we're going to start with acetyl CoA. And specifically, we're going to start with 2 acetyl CoAs. So the 2 reactants that start ketone body synthesis are 2 acetyl CoAs. And those acetyl CoAs will combine to form acetyl CoA. So pretty easy, right? You're literally combining 2 acetyls and you leave the CoA. The enzyme that catalyzes this conversion is called thiolase. The next step converts acetyl CoA into HMG CoA. The enzyme that catalyzes this conversion is an extremely important enzyme known as HMG CoA Synthase. So look at the name of the enzyme. It's synthasing or synthesizing HMG CoA. HMG CoA is written out and known as 3-hydroxy-3-methylgluteryl CoA. For the purposes of USMLE and COMLEX, you should be able to recognize that name, but you absolutely don't need to memorize it. So HMG CoA is what is formed by HMG CoA synthase. Now, it's important to note that in this step, you have to put in another acetyl CoA. So look how interesting this is, guys. You started with 2 acetyl CoAs and you formed acetyl CoA. And then in the second step, you took an already combined, basically double acetyl CoA, and you added a third. And once you did that, you formed HMG CoA. Now the next step is going to convert HMG CoA into acetoacetate, and the enzyme that catalyzes this conversion is HMG CoA lyase. And in this step, you actually have one of the products being acetyl CoA. So what's very important to notice, if we pause for a second and take a step back, is that the acetyl CoA is basically being recycled between steps 2 and 3. And this is very important in terms of feedback and driving ketone body synthesis. Now, what's very important to know is that acetoacetate is the first ketone that we've produced in this pathway, but it's not going to be the last. One of the extremely high yield points to keep in mind when you look and learn about ketone body synthesis is that you form ketones in a stepwise fashion. Sort of like in the catecholamine lecture when you formed one catecholamine before you could form the next, which were actually derived from the first catecholamine. The same exact thing is happening here in ketone body synthesis. So future ketones can be derived from acetoacetate, but acetoacetate is the first one produced. So here's where we are thus far, and this is a crucial step in ketone synthesis because once you form that first ketone, acetoacetate, you have 2 options. The first is that you can form another ketone called acetone, and the enzyme that catalyzes that conversion from acetoacetate into acetone is acetoacetate decarboxylase. So it's decarboxylating acetoacetate. So that helps you remember that the reactant is acetoacetate. And when you form acetone, you'll also form carbon dioxide, shown here in pink. The other option for acetoacetate, if it goes to the right in this diagram, is to form another ketone known as beta-hydroxybutyrate. And the enzyme that catalyzes this conversion is beta-hydroxybutyrate dehydrogenase. So here's where we are. At this point, everything you see here in green are ketones. Acetoacid is formed first, and derived from acetoacid are two other ketones known as acetone and beta-hydroxybutyrate. Now we'll get into a further discussion later in this video in terms of what the significance is of these different ketones and where they can be found in the body and what kind of symptoms they manifest, but it's extremely high yield to know, once again, that acetoacetate is the first ketone from which acetone and beta-hydroxybutyrate ketones are derived. So this is ketone body synthesis. This is the only pathway that you need to know. The enzymes are shown in red. The special products, reactants, what have you, are shown in blue and pink. And then along the way, you see in black letters the reactants and the products of the basic pathway. So the next thing that we need to talk about, which is an extremely, extremely important and high yield discussion, is when is ketone synthesis used, right? Under what conditions will the body want to form ketones? The first one is starvation. And the pathophysiology here is gluconeogenesis-induced depletion of oxaloacetate. And I'm going to show you a diagram that will explain this, simplify everything, and help you memorize this really high-yield concept. So in starvation, you have gluconeogenesis-induced depletion of oxaloacetate. Now, to remind you, this is what gluconeogenesis looks like, if it's very, very simplified. You start with pyruvate, you convert the pyruvate to oxaloacetate, and then you go back up all the way back to glucose. So this is basically the opposite of glycolysis. See the glycolysis versus gluconeogenesis lecture if you need more information. But this is what gluconeogenesis is. Again, it's forming glucose from pyruvate. And the first step is converting pyruvate into oxaloacetate. So what happens in this state of starvation? Well, when you're starving, the body wants glucose. So what it's going to do is it's going to convert pyruvate to oxaloacetate and go all the way back up to glucose. Now, in gluconeogenesis, when it's occurring over repeated amounts of time, the oxaloacetate will be greatly reduced. Because think about it, you're converting pyruvate to oxaloacetate, and then that oxaloacetate is going up to glucose. So over time, in prolonged periods of starvation, you deplete all of your oxaloacetate. Now, what happens next? Well, shown here on the right is the TCA cycle. And remember that the first step in the TCA cycle is to combine acetyl-CoA with oxaloacetate. But what happens if you've been going through gluconeogenesis repeatedly over the course of hours or days due to starvation? You're not going to have any oxaloacetate. So because oxaloacetate is depleted, the TCA cycle cannot work. It absolutely cannot work. Now, if the TCA cycle cannot work and you can't produce energy from the TCA cycle, then the body needs another fuel source. And that is where ketones will be formed, because ketones can act as fuel when other fuel sources cannot. So again, just to quickly summarize what starvation really means for ketones, in prolonged periods of starvation, you deplete all of your oxaloacetate because you're going through gluconeogenesis repeatedly. Once the oxaloacetate is gone, there's no action at the TCA cycle. Once there's no action at the TCA cycle, you obviously cannot form the intermediates that are required for the electron transport chain. And the body is going to need some other type of fuel besides glucose that doesn't really cost a lot of ATP. It can't get ATP from the electron transport chain because the TCA cycle doesn't work, and it can no longer get more glucose because we've exhausted all of our oxaloacetate, which not only prevents us from getting more glucose from gluconeogenesis, but it prevents the TCA cycle from working as well. So this is a very complex interplay between the TCA cycle and gluconeogenesis, as I'm sure you can now appreciate. High yield, bottom line, is that in states of starvation, oxaloacetate gets depleted due to repeated attempts to go through gluconeogenesis, which means not only can you not form glucose for fuel, but you can't go through the TCA cycle and ultimately the electron transport chain to get more ATP. That is what starvation is. Now, in a similar state, known as diabetic ketoacidosis, pretty much the same exact pathophysiology is taking place, and I'm going to prove it to you here. So I'm just switching the title from starvation to diabetic ketoacidosis. And in a nutshell, diabetic ketoacidosis is a period of starvation for type 1 diabetics. So let's talk about what happens in DKA and prove to you that the same exact pathophysiology takes place. So diabetic ketoacidosis, aka DKA, is still gluconeogenesis-induced depletion of oxaloacetate. So let's consider for a moment what happens in DKA. So a type 1 diabetic has a blood sugar of 600 for, let's say 5 or 6 hours. So very high blood glucose, known as hyperglycemia. What happens in states of hyperglycemia in a type 1 diabetic? Well, the reason that they're hyperglycemic is because they don't have insulin. So insulin levels are down. In a normal person, your body would compensate and produce insulin, but in a type 1 diabetic, the beta cells of the pancreas cannot secrete insulin, and therefore insulin levels are low. Now, you should know by now, after our thorough discussion in this biochemistry series, that whatever insulin does, glucagon does the opposite. So if insulin levels are low, then glucagon levels are high. Now consider for a second what happens to glycolysis and gluconeogenesis under these conditions of low insulin and high glucagon. Because insulin is down, glycolysis is not taking place. And because glucagon is elevated, gluconeogenesis is taking place. So over time, due to profound hyperglycemia in DKA, low levels of insulin will inhibit glycolysis, and elevated glucagon levels will promote gluconeogenesis. So if we take this set of conditions and sort of slide it to the bottom of the slide, let's look at what happens in gluconeogenesis and the TCA cycle. And it shouldn't surprise you that this is going to be identical to periods of starvation. So again, elevated glucagon will drive gluconeogenesis. So pyruvate will be converted to oxaloacetate. Oxaloacetate will go all the way up to glucose, and over time, the levels of oxaloacetate will be very much depleted. Because oxaloacetate is shot, there is no action at the TCA cycle because you cannot combine oxaloacetate with acetyl-CoA. Therefore, you cannot spin through the TCA cycle. Therefore, you cannot make the intermediates, which will ultimately go to the electron transport chain to ultimately form massive amounts of ATP. So to summarize, DKA is the same gluconeogenesis-induced depletion of oxaloacetate. The reason here being there are decreased levels of insulin and increased levels of glucagon. So DKA and starvation are functionally identical when it comes to the reasons that they form ketones. So when is ketone synthesis used? Remember, that was the question that we were considering before we had this discussion. And again, I'm going to use a chart to summarize everything that we've talked about so far. So first condition is prolonged starvation, and the pathophysiology is gluconeogenesis-induced depletion of oxaloacetate. And because oxaloacetate is gone, you cannot form glucose, you can't run the TCA cycle, and you can't make ATP. So the body says, give me them ketones. It needs some fuel source to keep the brain active. The second and functionally identical situation is diabetic ketoacidosis. Again, it's gluconeogenesis-induced depletion of oxaloacetate. In this case, insulin down, glucagon up, drives gluconeogenesis, depletes oxaloacetate, can no longer make glucose, can no longer run the TCA cycle. Body says, give me them ketones. So memorize that. It's extremely high yield. And now we're going to talk about the third and final situation, which is extremely high yield when ketones are synthesized. Now, this third and final state is alcoholism. And the pathophysiology here is alcohol-induced NADH to NAD plus ratio. So in other words, you're increasing the ratio of NADH to NAD plus. And I'm going to explain exactly why that is a problem and why the body needs ketones to compensate. Now, it's extremely important to understand how alcohol, or how ethanol, is metabolized. And there's a separate video in the Dirty Biochemistry series about alcohol metabolism. And see that video for a full discussion of this pathway. But in a nutshell, alcohol, also known as ethanol, is first broken down to acetaldehyde, and then acetaldehyde gets broken down to acetate. This is how the body metabolizes alcohol. And what's extremely high yield to know for the purposes of ketone synthesis is that in the first step of this pathway, when you convert ethanol to acetaldehyde, NAD plus gets converted to NADH. So in somebody who is an alcoholic who's constantly introducing ethanol into the body, you're going to have to metabolize loads and loads of ethanol. And over time, that will lead to a decreased amount of NAD plus and an increased amount of NADH. Now, normally, the body has a perfect balance between NAD plus and NADH, but in your alcoholics who constantly are introducing ethanol into the body, there is an exorbitant amount of NADH because in the process of breaking down all of that unnecessary alcohol that they are drinking, you're forming NADH and you're depleting NAD+. So to summarize, this is what you find. Increased ethanol leads to decreased NAD+, and increased NADH. So how the hell does this make the body form ketones? Well, let's refer back to the TCA cycle. In the TCA cycle, we already discussed that you form a product by combining acetyl-CoA and oxaloacetate. What's extremely important to know, and you can see the TCA cycle video in the Dirty Biochemistry series for more information, but in the formation of oxaloacetate, in the step where oxaloacetate is formed, you convert NAD+ to NADH. So you cannot form oxaloacetate unless you convert NAD+ to NADH. Now think about what we already said. In states of alcoholism, there is decreased amounts of NAD+, and effectively, the amount is 0. So if you knock out NAD+, then you can never form oxaloacetate. If you can never form oxaloacetate, not only can you not do gluconeogenesis and form glucose, but you can't do the TCA cycle. So this is very similar pathophysiology to what we've already been talking about. Some deficit in the body knocks out the ability to make oxaloacetate, which prevents you from doing gluconeogenesis and forming glucose, and prevents you from spinning the TCA cycle and forming the intermediates required to do the electron transport chain. So the body says, damn, I'm going to have to use ketones. And that is why ketone synthesis occurs in states of alcoholism. It's a high yield point that we should also bring up now. In alcoholics, they can actually have baseline levels of hypoglycemia. And the reason there is because if you knock out the ability to make oxaloacetate because you're depleting your NAD+, then you can't do gluconeogenesis, so your blood sugar is going to be a little bit lower. So this is why some alcoholics have hypoglycemia. So in summary, this is what we see in alcoholism. Now, how do you remember this? I'm going to give you a cool mnemonic. So because the problem with alcoholism is an increased ratio of NADH to NAD+, the way that I remember this is by thinking about the beer Natural Light. So you've probably heard people refer to this beer as Natty Light, and what I think of is NAD-E Light, NAD for NAD+. So Natty Light, if your alcoholics like Natty Light, then the problem with alcoholism causing ketones to be formed is because you're depleting your NAD+, or you're increasing your NADH, and either way, it's Natty Light. So for completeness sake, here is our chart. We're going to add alcoholism, and the pathophysiology is the increased NADH to NAD+ ratio, again, due to the fact that we are depleting NAD+ when we metabolize ethanol. So here is the pathway that we've talked about. This is ketone synthesis. A couple other high-yield things that we need to mention. First, like every biochemical pathway, you need to memorize the rate-limiting enzyme. So in ketone body synthesis, the rate-limiting enzyme is HMG COA Synthase, shown here in bolded red letters. So again, that's the enzyme that converts acetyl-acetyl-CoA into HMG CoA. So the rate-limiting enzyme is HMG CoA Synthase. So how do you remember this? Well, instead of HMG synthase, my mnemonic is to think, what is the purpose of ketone formation? So ketones are formed in periods of starvation. So I think of someone who's starving, and instead of HMG, I kind of think of OMG, and people sometimes go, oh my God. So HOMG, I'm starving, for HMG synthase. So the S in starving for synthase, and the HMG really is HOMG, or just OMG if you would prefer. But again, the reason that I remember this mnemonic is because HMG synthase is the rate-limiting enzyme of ketone synthesis, and we do ketone synthesis in periods that are functionally identical to starvation. So starvation, DKA, alcoholism, and I think of, oh my God, I'm starving. And for your pleasure, this is a picture of Adam Richman, the guy who did Man vs. Food. So here again is our summary of the biochemical pathway. Everything that you need to know in terms of ketone synthesis is shown here on this slide. Let's just wrap up by talking about some quick other high yields that you might see on your exam, and I'll give you some cool mnemonics to remember all this stuff. So the other high yields that you should know is that red blood cells cannot use ketones. And the way that I remember this is that RBCs are really basic cells, and really basic cells can't use ketones because they're just so basic. Second point is that urine ketone dips only detect acetoacetate. So we have dipsticks that you can dip in someone's urine, and you can see how many ketones are in the urine. This is particularly useful in type 1 diabetics who want to see if they're in diabetic ketoacidosis or not. But the thing here, and that's what's really high yield to know for USMLE and COMLEX, is that the urine ketone dipstick only detects acetoacetate. It doesn't detect acetone, and it doesn't detect beta-hydroxybutyrate. So the way that I remember this is that instead of acetoacetate, I remember acetoacetate and P for urine. So I remember that when you do a urine dip, you only can detect acetoacetate. So remember acetoacetate. The third and final high yield is that in a state of ketosis, or in any of these starvation periods, but especially in DKA, the person's breath will smell fruity, and the reason is because they're actually breathing out acetone. They're breathing out the acetone, and that has a fruity odor to it. So keep that in mind. It's extremely high yield. But this is it, guys. That's everything that you need to know about ketone synthesis. To summarize and to close really quickly, remember the pathway, remember the rate-limiting enzyme, really try to understand why DKA, starvation, and alcoholism leads to ketone synthesis in these periods of starvation where the body needs alternative fuel source because it can't make glucose and can't do the TCA cycle. It all boils down to the inability to get oxaloacetate into the body in some states because you're depriving the body of the oxaloacetate as you do more gluconeogenesis, and in others because you don't have the NAD+ necessary to actually create the oxaloacetate to begin with. But that is everything that you need to know about ketone body synthesis.